Welcome everyone to our second Ciao summer seminar. I am Clemens Wittwehr from the European Commission's Joint Research Center, where I am working in the Ciao project. The acronym Ciao is not only an Italian greeting, but it also stands for Modeling the Pathogenesis of COVID-19 using the Adverse Outcome Pathway Framework. My colleague, Dr. Brigitte Landesmann, and I will lead you through today's Ciao summer seminar, which will cover the topic of vaccines. As today's speaker, we have the privilege to welcome Dr. Florian Krammer. Dr. Krammer is Professor of Vaccinology at the Department of Microbiology at the Icahn School of Medicine at Mount Sinai in New York. And since 2019, Professor Krammer is the Principal Investigator of the Sinai-Emory Influenza Vaccine Innovation Center, which aims to develop improved seasonal and universal influenza virus vaccines. The Krammer Laboratory is part of the NIH-funded Centers for Excellence in Influenza Research and Surveillance, which focuses on understanding immune responses against RNA viruses such as influenza, with the goal to develop better vaccines and novel therapeutics. So when the COVID chaos hit us all, Dr. Krammer hit the ground running, and he immediately became one of the world's leading experts in SARS-CoV-2 antibody and vaccine research. Not only does he know more about the topic than anybody else, as German virologist Dorothee van Leer put it, he is also on the forefront of conveying his knowledge to a broader public in terms, as he said in a recent interview, which his grandparents should understand. So join me in welcoming Dr. Florian Krammer for today's Ciao summer seminar. Florian, the floor is yours.
Thank you very much for this very nice introduction. I doubt that I know everything about COVID-19 and vaccines, but what I'll try to do today is I'll try to give a bigger picture overview of where I see vaccines right now for SARS-CoV-2 and what I see for the next few months and maybe next few years, where this is moving and where SARS-CoV-2 might be moving. Okay, so I changed the title slightly to really focus this more on SARS-CoV-2 and COVID-19. And before I start, I wanted to just put up my disclosures. Our university has filed IB on SARS-CoV-2 serological assays and certain types of SARS-CoV-2 vaccines. I'm not really going to talk about any of those, but I think it's important to be transparent. All right, just to kind of review the situation that we have right now. What you can see here is the global number of infections and deaths as of today. We had about 550 million official infections and about 6.4 million deaths, official deaths so far. On the left side, you see, and you're probably very familiar with that, the epi curves. This is again globally, cases per day. And we can see that we had this recently, this huge BA1 Omicron wave, then a BA2 Omicron wave, and now we are gearing up towards, it seems, BA4, BA5 Omicron wave. The deaths are shown on the right side, so there were a lot of deaths, specifically in this winter season of 2021, but also the BA1 wave of Omicron caused a lot of deaths. Afterwards, the cases were falling, and they're actually right now at the lowest point they were ever in the pandemic, which is something that I find really nice, and I hope it stays like that, even though the BA5 cases increase. The problem is that the true number of deaths is likely substantially higher. There's two studies that have looked at that, with looking at the excess deaths, and one was basically put online by The Economist. The other one was published in the Lancet, and they arrive at about 18 million COVID-19 deaths by the end of 2021. And so this is probably much higher by now. But this is, of course, higher than the official numbers. On the plus side, we had a lot of countermeasures that were put in place, and I think the most consequential countermeasures were actually the vaccines that were developed. What you can see here on this slide is a number of vaccines that are used globally. It's not inclusive of all the vaccines that are used. There's other ones that have been licensed now as well. But at least in Europe, the big players are the mRNA vaccines by Moderna and Pfizer. You see that here on the left side. Initially, a lot of the vectored vaccines from AstraZeneca and J&J were used, not so much anymore. And now in Europe, we also have a licensed vaccines by Novavax, which is a recombinant protein vaccines, and inactivated vaccines are now also coming. If you look at this from a more global perspective, I have to say that the inactivated vaccines that were produced in China and in India actually made the biggest impact on global immunization. They were used in the largest number, far more than Moderna or Pfizer. I think that's something that we should keep in mind. But in Europe, of course, right now, most vaccines that are used are mRNA vaccines. For most of these vaccines, 2 or 3 injections are required. The numbers that you see here in parentheses, that's the efficacy that was reported in the initial clinical trials. And of course, this was relatively high against the ancestral strain, the original SARS-CoV-2. This changed a lot, unfortunately, by now. We hear a lot about waning and breakthrough infections and so on and so forth, but I really wanted to show some data, and this is from my laboratory and in collaboration with Viviana Simon, who is a colleague of mine here at Mount Sinai. We have a cohort that we started in April 2020 where we follow healthcare workers. Initially, this was a cohort based on infections, and then, of course, people started to get vaccinated. And so we monitor the antibody titers, and this is shown here. So on the y-axis, you have antibody titer. This is measured by a binding assay. On the x-axis, you have distance from vaccination in this case. So here on the left side, we have the original, the initial vaccination. The first 2 doses were given around here. And we have 2 groups here. In orange, we have people who had an infection before they were vaccinated and then got vaccinated. In blue, we have people who only got vaccinated, so they never had an infection before. And so we see initially the kinetics are a little bit different, but for both groups, when they get vaccinated, their titers shoot up. The blue group actually benefits a lot from the second shot. The orange group doesn't really. And then you see the absolute titers are higher with the orange in the orange group, in a way, because they got exposed 3 times to antigen, and the blue ones only 2 times. And this difference in quantity of antibody actually is maintained over time, but the kinetics of waning in this case are very similar, right? So you have this really high peak right after vaccination. These are antibodies that are produced by plasmablasts, which are short-lived B cells that die after about 10 days. The antibody stays in circulation, but IgG has a half-life of about 21 days, so they start to decline. And then we get to the stabilization phase here, which is basically driven by long-lived plasma cells in the bone marrow. So the titers really become stable after they wane about 10 to 15-fold. And then here on the right side, we have the situation after the booster vaccination. The booster vaccinations here were given on day 0. And you see this difference between the orange and the blue group is maintained here. But then once they get the booster, this is kind of an equalizer. Now both groups have similarly high titers. And what also stands out here is that after the 3rd vaccination, the kinetics are very different, right? You don't have this rapid waning anymore. You have some waning, but it's very gradual and it's very slow. And so looking at this, you could argue that the problem is really not waning of immunity because it stabilizes. It might actually be really the variants that changed and now escaped these wonderful immune responses, right? You're just not vaccinated against what is circulating anymore. So I don't actually think that waning of immunity is the big issue that we are facing. Big issue is, I think, here. So this is an overview of variants of concern. And so at the same time, or actually a little bit before the vaccines were rolled out, variants of concern were detected. This was something relatively unexpected for SARS-CoV-2 because coronaviruses have proofreading activity and they're relatively stable. But the first 2 variants that were detected were detected because they were growing faster than the ancestral strain in the population. They were taking over the infections. And there's still an argument going on if B117 or the alpha variant was first detected and first found, or if it was B1351 in South Africa that was reported first. It's not really clear. But with alpha, we saw that there was an amino acid change in an important interface between the receptor binding domain here on the spike protein and ACE2, the receptor. And so this change from N to Y increased the affinity for the receptor, and that probably made the virus more transmissible, more infectious. And for the beta variant and also the gamma variant, which was then discovered in Japan and was originally from Brazil, we have additional changes in the receptor binding domain that they have this N501Y mutation too, but they have additional changes, and those actually mediate a degree of antibody escape. So those were really the first escape mutants, although the escape was only partial. And then we saw a lot of other variants of interest, not variants of concern, that came up: lambda, mu, epsilon, zeta, eta, theta, iota. Iota was our local New York variant. Kappa. They didn't really cause huge issues, and they disappeared relatively quickly. So none of these variants were actually that problematic from the vaccine point of view. The first one that really became problematic was then Delta, and this was in spring of 2021. Delta emerged during a large wave in India, and then spread globally. And the situation in spring of 2021 was actually pretty good, right? So case numbers were falling. This is from the US, but the same is true for Europe. People started to get vaccinated more and more, and the idea was it's going to be over soon. This is looking good, right? I also was under the impression that soon we will have it under control. But then Delta started to circulate globally and took over. And here in the US, basically within a few weeks, basically 8 weeks, it started to dominate, and then caused a huge summer and fall wave in the United States, but also in Europe, and actually led to a lot of deaths. The problem here was not so much that there was a strong escape mutant in terms of the mutations that it has on the spike, but it had a very high replication efficiency in the upper respiratory tract, and was very, very infectious. And I think that was more the problem than really escaping from neutralizing antibodies. And then I won't go through all of this, but in November 2021, Omicron was detected. The first sequence came from Hong Kong, from a traveler who was quarantining there. The traveler came from South Africa. The sequence was put online on November 22nd. People were already looking at the sequence and ringing the alarm bell. A day later, there were more sequences from Botswana and South Africa. The problem here was that there was a huge amount of mutations in the spike protein that covered basically all neutralizing epitopes. And then within a few days, this became a variant of concern, and basically then spread within 4 weeks globally. And in mid-December, we already had a huge Omicron wave here in New York City. And here on the left side, you see the spike protein of Omicron, and all the mutations are indicated. This here is actually BA1. BA2 was detected at the same time, and we have at least, we have 15 mutations in the receptor binding domain, which is a huge amount of mutations, very unexpected. But of course, this had a lot of consequences, and the NTT was also heavily mutated, and there's additional mutations throughout the spike protein. But what that meant was that the virus was able to escape neutralizing antibody responses to a large degree. And this is shown here on the right side. These are neutralizing titers for different groups of people. We always looked here at neutralizing activity against the wild type virus, then beta, which was the worst case scenario until Omicron in terms of escape. And then here is B11529 or Omicron. Again, this is BA1. Here we have sera from convalescent individuals who initially got infected with the wild-type ancestral strain, and you see there's a huge drop in neutralizing activity. Actually, more than 70% of individuals don't have neutralizing activity against Omicron at all. And this looks similar for people who got vaccinated twice with an mRNA vaccine. There's this huge drop, 30% of people lose all their neutralizing activity. If you got vaccinated 3 times, or if you have hybrid immunity, meaning an infection plus 2 vaccinations, it looks a little bit better. There's still a huge drop, but because people have high titers to begin with, they have more residual neutralization activity against Omicron. And of course, this had consequences. There were a lot of breakthrough infections. Even 3 shots of the Pfizer vaccine or the Moderna vaccine only have residual protection against infection. But of course, the protection against severe disease is still relatively good. The other problem with Omicron is that, of course, as you all know, we have a lot of subvariants. What was initially detected was BA1, BA2, and BA3. BA3 really kind of disappeared and never caused a big problem. But after the big BA1 wave, we had a BA2 wave. In New York City, we had a subvariant of BA2 that was causing issues, BA2.12.1. And of course, now we are dealing with BA4 and BA5. And there's new subvariants that come up, including BA1.23. I will talk a little bit about that later. And then also BA2.75, which has additional mutations. And now we're also seeing more and more recombinants between those. And of course, these subvariants also have an impact. This is more neutralization data. This is now from a paper from David Ho's group at Columbia, where he looked at people who were boosted, people who had hybrid immunity, meaning infection and then vaccination, and then people who were vaccinated and had either BA1 breakthrough or BA2 breakthrough. And we can see clearly here that there is an additional drop from basically BA1 and BA2 towards BA4, 5, right? So the titers go down. The additional mutations that BA4 and 5 have impact further on neutralizing activity. This is a little bit less pronounced when you look at BA2 breakthroughs, and that makes sense because BA2 and BA4, 5 are closer related. But this suggests that probably reinfections can happen, and that even if you had a BA1 breakthrough, that your immunity against BA2, against BA4 and BA5 is really suboptimal. But the other thing that we have to keep in mind here is that the vaccines still do a pretty good job in protecting from severe disease. So what I'm showing here is data from New York State. New York State measures vaccine effectiveness data every week. And they look at vaccine effectiveness against mild or any symptomatic disease, and they look at hospitalization. This is not stratified into different vaccines or 2 shots versus 3 shots. That's just the overall population. And what they see here is that, of course, initially the vaccine effectiveness against any symptomatic disease was relatively high, then started to fall. This is here the impact of Delta. We're going down to about 80%. And then with the arrival of Omicron, this is further dropping. Here, this goes till March only, but it goes down to about 70%. The good news here was that for the vaccine effectiveness against hospitalization, starts at about 95, between 93 and 95%, fluctuates, but basically even with Omicron, we are still at the 90%. And there are different data sets for this out there. Some see more of a drop, some see less of a drop. But even if you look at the Kaiser Permanente study from California or Oregon, you still see that the protection against severe disease is relatively well maintained. And the real example for showing this contrast is Hong Kong versus New Zealand. So Hong Kong and New Zealand are both countries that kept the virus out for a long time, and they were really good at managing the situation, right? But then with Omicron, the virus made it into Hong Kong, it made it into New Zealand, and there was no way to keep it out anymore. And this is shown here on the left side. These are basically cases normalized per million people. In blue, we have Hong Kong, so there's a very steep curve, and then case numbers drop again. And then we have New Zealand in green. So the curve is not as steep, but it's also a lot of cases, and then this drags on a little bit. It's not going down as quickly as in Hong Kong, but overall it's comparable. And here on the right side, you see the deaths. Again, this is normalized per million people, and you see this huge wave in Hong Kong, but you see very few deaths in New Zealand. And the question is, what's the difference here? The difference is basically vaccination. In Hong Kong, they managed to only vaccinate about 30% of their 80+ population. And in New Zealand, it was way above 90% in terms of vaccination in the 80+ population. And that really made this difference. People get infected, they get sick, but they don't necessarily die, or they die at a much lower rate if they have this baseline immunity from vaccination. Switching gears a little bit, how will SARS-CoV-2 evolve? And that's an interesting question. And there's a lot of very interesting aspects here that are super important for vaccine development. So typically for viruses, and credits to Jesse Bloom here, who gave me this slide, typically for viruses, we see different patterns of evolution. What you often see is this ladder-like evolution, where you have one variant and it mutates a little and becomes the next variant, and mutates a little and becomes the next variant. We see that for common cold coronaviruses. We see that for different influenza A viruses. And this is something relatively easy to deal with, right? Of course, you might have to update vaccines eventually, but it's hard to go wrong here. In some cases, there are lineage splits, right? We have that for influenza B, where we have two lineages. OC43, a human common cold coronavirus, did that as well. There's two lineages, but in general, this is easy to deal with too. You might need a bivalent vaccine to target this, but you know, it's still not a big deal. The problem is if you have a very chaotic evolution, and this is something that we have seen for H3 and 2, for example, before the pandemic, where we have many different clades that co-evolve, they exist next to each other, and it's really hard to target those with vaccination. Now, if we look at SARS-CoV-2, it's even worse. We have this star-like phylogenetic tree, and all these variants are actually coming out of the center. They're coming from wild type so far. So Omicron is not a descendant of Delta. Delta was not a descendant of Alpha or Beta. So they're all coming out of the center. They're all coming from wild type. And that makes it really hard to predict evolution. Now we see a little bit of a different story with Omicron, where, of course, BA4, BA5 came out of BA2, right? But in general, that's not what we observed before. And that makes it really hard to predict, because the next variant might come out of the center again, or it might come out of Delta, for example. And to dig a little bit deeper, why we are seeing this, we have to talk a little bit more about what's happening in immunocompromised hosts. So first of all, coronaviruses are an exception among RNA viruses, right? A lot of RNA viruses make a lot of mistakes and have a high mutation rate because they don't have proofreading activity when they copy their genome, right? There's a lot of mistakes and they don't get corrected. Now, coronaviruses do have proofreading activity. They can correct their mistakes. And that's why they mutate usually slower. Now, the problem is if you have a lot of infections, like in the pandemic, and you have a wave, then you have a lot of replication cycles, right? In every infected person, we have a lot of replication cycles. And each replication cycle in each cell allows the virus to explore space and look at, try different mutations, right? The more replication you have, the more chance of mutations you have. That's one problem. But the other problem, and I think that's even more significant, is that we see faster evolution in immunocompromised hosts. And this is something that has been seen in the beginning of the pandemic already. People can get persistently infected with SARS-CoV-2, and this is usually people who are immunocompromised. This is from a New England Journal of Medicine paper here, where somebody got infected and was infected for 150 days. This was an immunocompromised patient, and the virus was isolated at different days and then sequenced. And what we see here coming up are mutations that we found in variants, right? E484K, for example, that's in the beta variant and in the gamma variant. N501Y, that's in the alpha, beta, and gamma variant, right? So these mutations were coming up over time. And the idea is that immunocompromised people who cannot get rid of the virus, but build some type of immune pressure on the virus, and that really kind of drives the evolution of the virus in that person. In addition to that, you might actually have different strains that develop in that person, and then coronaviruses can recombine, and that might be an additional mechanism why we see a lot of these mutations. And this is just an example from our hospital. Again, this is work done by my colleague Viviana Simon. And so they basically saw this happening. They saw basically the emergence of a variant in real time. So this is this is data from a patient who got infected in December with BA1, with Omicron, and was infected for 4 months. And over time, this patient was sampled every now and then. And in the end, we saw that there were 8 mutations in the spike protein. And that's, of course, concerning, right? But in addition to that, this virus was transmitted onto other patients. And even worse, the CDC detected the same virus afterwards in the population in New York City. So this is a fit virus that was actually spreading in our population in the city, and it might still be spreading. And these additional mutations, of course, allow it to even more escape from the antibody response. This is just a phylogenetic tree, basically showing here the initial case over time in red, and then these other patients that got infected, and then here the GSET numbers. These are 2 cases that were picked up from the population. So this is really a big problem, and this is an issue because we assume that there is 10,000s of people out there that have these persistent infections, and each of them could actually develop a new variant. And so, in my opinion, the potential that we get variant after variant after variant is certainly there. But again, when we now look at all the baseline immunity that people have through vaccination, unfortunately also through infections, we have to kind of ask ourselves, what's a realistic long-term scenario for SARS-CoV-2? And I think it's worth looking at other seasonal viruses, seasonal respiratory viruses for that. And so I think we have 2 scenarios there, and SARS-CoV-2 might actually fall somewhere in the middle. On one side, we have human coronaviruses that circulate seasonally. Usually you get more and more cases in fall and then in winter. We have relatively mild disease, and because of that, we have no vaccines. So this is the best case scenario, I would say. And then on the other side, we have influenza, which is, in my opinion, the worst case scenario, where we also have seasonal circulation. We have a substantial degree of severe disease. There's a lot of people who get very sick. There's actually a lot of people dying of influenza when there is a proper influenza season. And because of that, we need annual vaccination. And right now, it's not really clear over time where SARS-CoV-2 might fall. I think right now it's much closer to influenza, but if you follow season after season after season, it might actually get closer to human coronaviruses. Not because I think the virus is becoming milder, just because people build up more and more immunity. But even with these scenarios, there's a lot of issues that are currently remaining that are problematic. The first one is we don't know how to prevent infection in mild disease well. Initially, the vaccines did that, but they're not doing it anymore, which again, probably has more to do with the virus changing than with really waning immunity. And that's a problem because if you get infected, if you have a breakthrough infection, you still have a good chance of developing long COVID, unfortunately, and long COVID is a real issue. And so I think it would be important to invest into making vaccines that prevent infection and mild disease. Also, this is important from another point of view. If you have less virus circulating because people are protected from infection, it's easier to protect people who cannot get protected by vaccination, right? To protect vulnerable populations, because then they have a lower chance of even getting infected. The next question is, how do we deal with these emergent variants, right? How do we keep up? And then the last point is really how do we protect individuals who cannot be protected by vaccination, people who have a compromised immune system. And this is an issue with SARS-CoV-2 for sure, but this has also been a long-term issue for influenza, for example, and for other viral infections. So that's not new, but I don't think we have good solutions for it. In terms of the protection, we also have to think about what we want, right? Currently, what we see with the vaccines is protection from severe disease, which mechanistically often comes from an anamnestic B and T cell response, non-neutralizing antibodies. That's where we are right now. But what we would like to have is protection from infection. And for that, you need high titers of specific neutralizing antibodies, and especially on mucosal surfaces. So that's where we want to end up. And one of the issues that I see with the vaccines that we develop is that there's, in a way, a design flaw there. They were not designed to protect us from from infection. They were designed to protect us from disease. So if you think about immune responses after respiratory tract infection, right? So you get infected, you have virus in the upper and partially probably also in the lower respiratory tract, and your immune system responds. And typically it responds with a systemic immune response that includes a lot of IgG in serum, and that IgG ends up also in the lung and protects us from severe disease when we get reinfected. But you also get secreted IgA in the upper respiratory tract. And that requires local stimulation, which happens during a respiratory tract infection. And if that mucosal antibody can neutralize the virus that you're exposed to, then you never develop an infection, right? Because the virus lands on your mucosal surface as it's getting neutralized, and you just don't get infected, right? And that's the ideal scenario. Of course, if the virus changes a lot, then you can have these mucosal antibodies, but they still can't prevent you from getting infected. But hypothetically, if the strain is still recognized, they can. Now, what we do with intramuscular vaccination is we get a lot of this systemic IgG that protect our lung, but we don't really get a lot of mucosal immunity, right? Initially, and you've seen when these titers shoot up after vaccination, there's some IgG that ends up on mucosal surfaces. And that's probably why initially we were also protected from infection. But then as these titers wane 10 to 15-fold, there's very little IgG on these mucosal surfaces left that can neutralize the virus. And so one solution to that would be intranasal vaccination. Intranasal vaccination basically mimics natural infection and gives you also this mucosal immune response in the upper respiratory tract, and hypothetically that can protect us from infection. And so this is something, I think this is a space that we need to follow. The problem that I see is that we are quite far away from having a mucosal vaccine against SARS-CoV-2. And I put here a list of vaccine trials. This is now, I think, a month old or so, with vaccines, with mucosal vaccines that are developed against SARS-CoV-2, right? A lot of them are applied intranasally. Some of them are even given orally or sublingually. And a lot of them are in early clinical trials, specifically the ones that are developed in the UK, in the US, somewhere in Europe, are in early clinical stages. There are some that are further, that are in phase 3 trials. but this is typically outside of the European Union, including in India and in Iran, for example. And even if those vaccines work, and some of them might actually work relatively well, I'm specifically counting here on the vaccine that Bharat is developing. Even if they're working, it might be hard to get them licensed in the EU. So that's why I'm saying that these vaccines might still be quite far away. The other point is we need variant-specific vaccines, right? It doesn't help us if we have antibodies against the ancestral strain, but they don't neutralize our currently circulating variants very well anymore. And so I think it's really important that the vaccines get updated. And there were news, basically, very recently, Moderna had a press release saying that they have positive data with their bivalent vaccine that contains BA.1 and the ancestral strain. And at the VRBPAC meeting on June 28th, they showed that data to the FDA. Pfizer also had data. I'll walk you through some of the data sets. And it looks like there might be the variant-specific vaccines coming for fall. The application packages were filed with the FDA, but also with the EMA. And the question is just now, how long is it going to take until they can be used in patients? And the main problem, and I'll talk a little bit more about that, is that right now we don't have a quick pathway for licensure of these updated vaccines. And I think it's up to the regulatory agencies to design a good pathway to do that. And I'll propose a few things as well. So this is the data from the Moderna press release and what they also showed at the VRBPAC meeting. What they have here is they have individuals who now got a booster dose. You see the pre-booster titers against Omicron and the post-booster titers, and they got a booster dose that was either the ancestral mRNA vaccine or the bivalent vaccine that contains both the ancestral and the BA.1. And what you can see is that if you use the bivalent vaccine, you actually get higher titers against Omicron. Now, these are neutralization titers, but we also have to think a little bit about the epitopes targeted, right? So here, while you get this boost against Omicron, it's probably very few epitopes that can be targeted by these cross-neutralizing antibodies. Well, here you probably have a lot of epitopes that are targeted that are specific for Omicron, right? And so I think that's also an additional advantage here, even though the titers are not that much higher. They are significantly higher, not that much higher. And they see the same trend in people who were seronegative, meaning who never had an infection, but also in people who had a breakthrough infection. And Pfizer had similar data. They actually have more data. They did side-by-side comparison of a booster dose that had only Omicron or a bivalent one, which has ancestral and Omicron. And it's interesting and makes sense from an immunological perspective that if they only use Omicron, they actually see higher titers against Omicron. So again, they have a similar study design here. These are the booster doses that were given to the different groups. If you give the regular ancestral booster dose, you end up with a neutralization titer of about 450. If you give the Omicron specific one, it's about double. If you give a higher dose of the Omicron specific one, it's about triple the titer. If you give the bivalent ones, the titers are a little bit lower. And so I think this is really a way to move forward to use these adapted vaccines. Ideally, we should actually use BA.4, BA.5 sequences and not BA.1 sequences, but the data that has been generated so far was with BA.1. And so the situation so far with Omicron was that, you know, in November, Omicron was detected. Then we had massive waves with BA.1. We had BA.2 waves early in the year. Now we are running into BA.4-5 waves. And maybe in fall, we get a BA.1 vaccine that is rolled out. And when we look at an updated vaccine and the development, vaccine production started in December, January. The clinical trials started in February, March. Trial results were available basically by May, then were presented to the FDA in June. And so this takes a very long time. It's actually 11 months to get in a way, an outdated vaccine, because this is BA.1, to the market. And in my opinion, we need to kind of change how we do this and how we think about it. And so one way to do this much quicker would be a simple strain change without clinical trials. And this might sound outrageous, but if you do that, the vaccine would probably be on the market within, an updated vaccine would probably be on the market within 2 to 3 months. And actually, that's exactly what we are doing for influenza. For influenza, we just change the composition of the vaccine every year. That does not require clinical trials. That's just based on characterization of what's circulating. And we look if the titers drop against the circulating viruses, and if they do, we update the vaccine. So a similar system could be put in place for SARS-CoV-2. And the other question is, you know, when do we update the vaccine? Should that be an annual process, or should that really be done when a concerning variant emerges, right? A new variant. And when I'm talking about the concerning variant, I'm not talking about the difference between BA.1 and BA.2. I'm talking about the difference between Delta and Omicron, for example. And if you think about this, a vaccine that's rapidly available, that's matched to an emerging variant would have a maximum impact on the number of infections. And again, we're ending up with what model should we follow? Revaccination every year with an potentially updated vaccine, or revaccination based on new variants that emerge if they emerge. So that's another thing that we have to figure out. I know that we vaccinate every year for influenza, but that's really an exception. That's the only virus where we do that, and I don't actually think that's an ideal scenario. But in the long run, I think what we need to develop is variant-proof vaccines that basically cover all these antigenic space, where you wouldn't have to worry about a new variant emerging, where the vaccine would still work. And this could be multivalent vaccines, vaccines that focus on conserved regions of the receptor binding domain. There are some consensus sequence vaccines that are in development. And there are some good approaches, and some are in early clinical stage trials, and some are approaching clinical trials. The problem that I see is there's no Operation Warp Speed anymore. There's not a lot of appetite to develop these vaccines quickly, and there's certainly not a lot of money for doing that, right? And so it becomes, in a way, something that might drag out over years, because, of course, it's difficult to develop these vaccines and to get regulatory approval. Because the question is, how do you get a claim for variant proof if you can only show that the vaccine works against current variants, but you cannot show if it works against future variants yet, right? So there's a lot of regulatory and finance issues with this. And the other development that is ongoing and that I find interesting is universal coronavirus vaccines. People often put that term out, but there's actually a lot of different definitions and different things we need to think about. I think the easiest would be to develop a pan-Sabecco vaccine that covers all Sabecco viruses, of which SARS-CoV-1 and SARS-CoV-2 are members. That's still a relatively narrow part of the coronaviruses, and this might be feasible. People are working on pan-beta coronaviruses vaccines that also include Merbeco viruses, for example. That's where MERS-CoV-2 sits, which is another issue that we have with coronaviruses. And they could also cover human common cold coronaviruses, OC43 and HKU1. I think this is much more challenging and will take much longer. And there are even people thinking about developing truly universal coronavirus vaccines that would cover alpha, beta, gamma, and delta coronaviruses. I'm not talking about the variants here. I'm really talking about the different branches of the coronavirus family. And this is probably impossible, but on the other side, it's probably worth trying. And so in the end, I want to just come back to the most vulnerable populations, and that's immunocompromised individuals. And so I got very passionate about that because we had this one case at Mount Sinai where somebody, a multiple myeloma patient, got CAR T-cell treatment and then got vaccinated about 90 days later, twice with an mRNA vaccine, and then unfortunately got infected relatively quickly afterwards, was admitted to the ICU and died. And retrospectively, that person didn't make any immune response to the vaccine. And after that, we started to look into a larger multiple myeloma cohort and found that 15% of the people in that cohort did not respond to the vaccine at all. And if people responded, they had much quicker waning of immune responses. And so I think this is a really big issue that we still need to address, because we have a lot of people in our communities that cannot protect themselves. They don't have an immune system that's active enough to respond well to vaccines, and we have to do something to protect them, right? And there's a few things that can be done. One is, and we have participated in a lot of these studies now, to give a fourth dose. Some people who don't respond after 2 or 3 doses actually start to respond to the fourth dose. There might not be optimal protection, but you see at least some type of immunity developing, and that could help in the case of an infection. And this is also going hand in hand with what a study in Israel found. This was the first large-scale study looking at the effectiveness of the fourth dose of mRNA vaccine. This was done in an older population, I think 60 or 65 plus. What they saw was when you give a fourth dose of vaccine, there's some increase in protection from infection, 2-fold increase compared to 3 doses. And that's very transient, so it doesn't really help you against infection. But they saw that there was a more dramatic effect on protection from severe illness. And that's probably the same effect, that you have people in these cohorts that don't mount good immune responses after 2 or 3 shots, just because they're immunocompromised. But then with a fourth shot, you either give another stimulus to the immune system that now responds, or maybe they're off a certain treatment that they were on for the last 3 shots that suppressed the immune system. So I think for elderly or immunocompromised individuals, having a fourth dose right now, even if it's not the updated vaccine, is a good idea, specifically when we know that there is an increased number of cases with BA4 and BA5. And the other thing that, of course, changed the situation quite a lot was treatments, right? And so with Omicron, unfortunately, a lot of our monoclonal antibody treatments became obsolete, right? So Trovimab, or S309, was still working against BA1, but it's not working against the other subvariant very well. In the US, we have the luxury that we still have a monoclonal antibody on the market, Bebtelovimab, which works against all Omicron subvariants, even BA4 and BA5. And that is used in the US quite a bit to treat people who are at risk and who recently got infected. And then there's 2 small molecule drugs, molnupiravir, which is not very effective, but certainly has a measurable effect in reduction of hospitalization and death. And then, of course, Paxlovid, which you're all familiar with, which is the combination of 2 drugs that has a dramatic reduction leads to a dramatic reduction in hospitalization and deaths. And then, of course, there's also prophylactic antibody cocktail, Evusheld, which can be given months ahead. Has a very long half-life because the antibodies have been modified to give them a longer half-life. And actually, one of the 2 antibodies in Evusheld now works relatively well against BA4, BA5, and neutralizes that variant quite well. So we have a lot of tools to protect our immunocompromised populations. And this kind of brings us full circle, because if we cannot protect the immunocompromised, they will develop these persistent infections, and that will lead to more variants. And so I think it's really important to kind of take care of the most vulnerable and make sure that we have treatments for them to also make sure not just to treat them for their well-being, but also to make sure that the virus doesn't get a chance to evolve in them. And so to just summarize all of this, evolution of SARS-CoV-2 remains unpredictable, and the virus is certainly here to stay. It's not going away. We need updated vaccines, and even more so, more importantly, we need a regulatory pathway to update vaccines quickly. And this is right now a long process, but it needs to be possible to do that within 2 to 3 months. We need improved SARS-CoV-2 vaccines. Vaccines that give mucosal immunity would be ideal to inhibit infection or transmission. But I think we also need vaccines that give us broader protection. Variant-proof vaccines would be something that I think is feasible, and we should put a lot of energy into developing those. Universal coronavirus vaccines, I think that's a good approach in its different aspects, but I think those are requiring a much longer lead time and much more investment. And the last 2 points are long COVID is real. We don't understand it well. You probably heard a lot from Akiko about it and the mechanisms from David Petrino, but I think it's actually a whole complex of different mechanisms that lead to different symptoms, and that makes it really hard to treat. But because it's real and because it's doing a lot of damage, we need to really find a way to cut down on infections. And then the other problem that remains is really immunocompromised individuals who have a high risk, and by getting these persistent infections also put other people at high risk. So we need to find a way to protect them from infection, and if they get infected, we need to make sure that we can cure them right away. And I'll stop here, and I'm happy to answer any questions or participate in a discussion.
Wow, thank you very much, Florian. That was really great. What a wonderful overview of things that that happened, will happen, and are happening right now. Some bleak prospects, some optimistic prospects, but I think that's the summary of the story. I invite now every one of you, if you have a question, want to write it into the chat, please do so. We have one question from Julia, but if you want to ask your question in person, please just raise your hand and we will ask you to unmute yourself. So Brigitte, take over. You are the moderator of the question.
Julia, maybe you want to ask the question yourself that you wrote?
Yes, I could. Thank you very much for the opportunity. Thank you for taking the time to talk to us, Dr. Krammer. Some time ago, actually, when in the worst time when we were really locked and did a lot of reading, I read about outlined an approach where synthetic decoys that would mimic, there was an approach to mimic actually ACE2 itself, which I didn't think is a good idea because ACE2 has a very important function in the body. But then decoys to actually mimic the surface on ACE2, even in the nanomaterial form, to kind of get away the virus. And at that time, it wasn't even talked about local delivery. So I wonder, what do you know about it and what do you think about that approach? Because that would, I think, be also applicable to the immunosuppressed people, because it doesn't depend on developing immunity, but it would be kind of protective, or how should we say, prophylactic.
Yeah, I think it's an interesting approach. There are two key things that need to be achieved, and I don't think there was a lot of movement there. The first one is you need local concentrations of that wherever the virus is replicating, right? So just giving it intravenously is not going to work well, most likely. And the other problem that I see is, that's exactly what an antibody does, right? An antibody, a lot of antibodies bind to that interface, the receptor binding motif, where the RBD binds to the ACE2, right? And these antibodies are thrown off by the mutations that we see in variants, right? And so you would need to have to design this molecule in a way that doesn't happen. And of course, it doesn't happen with ACE2 because the virus still can interact with ACE2, but the antibodies are getting thrown off, right? So it is also a design question, how well you can do that in order to keep it variant proof as well, right? So I think it's an interesting approach, but I don't think there's anything that's close to getting on the market or anything that's even close to successful phase 3 trials right now.
I was thinking more of a local delivery rather than intravenous, but if I may, just a bit provocative question. Do you think that the idea of is one of the reasons potentially that it wasn't developed, that a lot of focus was put on the vaccines that were developed by the pharma industry?
No, I don't think so. There was actually there were a lot of initiatives, and I was involved in a few of those projects too, because, you know, there's a lot of drugs that bind actually to ACE2 and to that interface, right? And there was a lot of screening ongoing if any of those drugs might have an effect, right? I think there was also initially a concern that ACE inhibitors, when you take them for blood pressure, that they might have an influence on how disease looks like, either positive or negative, right? So I think there was actually a lot of interest, and if a pharma company would find that this is a potent way of protecting from disease, they would use that. And I mean, Paxlovid, for example, was developed also actually by Pfizer while they were developing a vaccine too, right? I don't think that's a reason, just because the vaccines were coming. That's a different, in a way, a different branch of approaching, or a different way of approaching the problem. They're usually not competing with each other.
Okay, we have got another question in the chat from Gelina. Is there a difference between natural infection and vaccination on mucosal immunity and the duration of protection?
Yeah, there's a difference. You don't necessarily get mucosal immunity when you get vaccinated, right? You get that from infection. We do see in some cases that people who got infected and then get vaccinated see a boost in mucosal immunity when they get the shot. And in terms of duration of protection, it's relatively similar. We see long-term protection with natural infection. We see that with vaccination as well. The real deal breaker here was Omicron. And in both cases, the protection waned quickly, or waned almost completely against Omicron, right? While both vaccination and or infection gave good protection against infection before Omicron. So there are different aspects involved, but I don't think one is better than the other. Except that if you get infected, you have a lot of risk of developing a problematic disease, right? Which is not the case for vaccination.
So there's another question. I've heard that from Helen. I've heard that some people who have had COVID lose a large amount of their sense of smell, and it doesn't come back. Would be a nasal vaccine also would cause a nasal vaccine also cause this, or could it prevent the effect of loss of the sense of smell?
No, it wouldn't cause that, and it would prevent that, because what happens is you basically get infected, and the virus destroys cells that the olfactory cells, right? And also the I forgot how these cells are called that sense taste. The virus really infects them directly and kills them, right? And if you have mucosal immunity, the virus can't do that, and you would be protected from that.
I also have a question. May I ask you a personal question on what the current knowledge? What would be your personal preference for the fourth shot in autumn? Which of the probably available vaccines would you choose?
I would probably take a monovalent BA.1 vaccine if this becomes available. My choice would be a BA.5 vaccine, but I doubt that we'll have that. And in terms of the platforms, I think either mRNA or the recombinant protein vaccines are pretty good, and I would take whatever I can get.
And what if it would be bivalent then, or only monovalent BA.1?
I think it will be different. I think for Pfizer it will be a monovalent, and for Moderna it would be a bivalent. Yeah.
But they say that the Delta might come back, and there might be a mix. Or how is the protection against Delta then if you have the booster for monovalent for BA.1?
I think that the protection against Delta is still very, very good after your 3 shots that you already have. I don't think you need another shot to protect you from Delta.
Okay.
So I'm not worried about that.
Are there any other questions from the audience? Can't see anyone. So our time actually is over, shortly over, 3 minutes past 5. So thank you very much for this very comprehensive overview. And Clemens, over to you
Yes.
again.
Yeah, thank you very much, Florian. It was really great having you. We will put a recording of this talk and the questions on our YouTube channel, on our Ciao YouTube channel. We will distribute the link as soon as it gets available. And in the name of the Ciao community and all the participants of today's seminar, I like to thank Florian very much for his time. And we are looking forward to more developments out of your lab, and we will keep a close eye on what's coming next.
Thanks for the invitation.
Okay, thank you. Goodbye.
Bye-bye. Thank you.
Have a nice day.
Thanks again. That's great.